Vegetative reproduction includes all those processes of propagation in which a part of the plant's body is separated from the parent and gives rise to a new individual without any obvious changes in the protoplast. Regeneration of new plants from parts of vegetative organs is called vegetative reproduction or vegetative propagation. Root better than hardwood cuttings. Care should be taken to put the morphologically lower side of the cutting in the soil. Roots are formed on the lower end of the cutting, and rooting is better if round and covered with moist soil in such a way that its growing tip remains above the soil surface. This bent branch is called a layer. After a few days, the covered portion of the stem usually produces adventitious roots. The rooted branch is then cut off from the parent plant and grown as an independent plant. The formation of adventitious roots in a layer can be hastened by injuring the layer by tonguing, ringing or notching. In tonguing, an oblique upwards cut is made on the stem. In ringing, a complete ring of bark, about 2 centimeters wide, is removed from the stem, whereas in notching, a V-shaped cut is given somewhere through the middle of the stem. Air layering technique, also known as goutte, is usually employed in those plants where stem branches cannot be bent to the ground. Pomegranate, orange, guava, lychee, etc. are the examples of such plants. The stem is girdled, that is, a ring of bark tissue is removed or slit at an upward angle and then covered with moist moss or cotton and wrapped with a polythene bandage to prevent drying. In drier climate, an earthen pot with a hole at the bottom is hanged over the bandage in a convenient position, and the two are connected by a soft cotton cord. The pot is then filled with water. Water, when trickles down the cord, keeps the bandage constantly moist. Auxins may be applied to the girdle region to promote the root formation. Adventitious roots are formed above the girdle portion. After rooting, the stem is cut off below the girdle portion and planted in the soil. Before planting, leaves are usually removed from the stem to prevent transpiration. The third way of artificial propagation is grafting. It is the process of joining together parts of two different plants in such a manner that they live as one plant. Grafting of plants is a widely used technique in horticulture to multiply the desired genotypes in mango, citrus, apple, pear, guava, among others. A portion of the plant is inserted into another plant of the same species or a compatible plant of different species or even genera. Of the two plants, the one rooted in the soil is known as stock and the other grafted on it as scion. When the cambium of the stock plant comes into physical contact with the cambium of the scion, both form new xylem and phloem simultaneously. Consequently, the stock and scion become united and grow as one plant. It is the scion which largely determines the type of plant one gets after grafting, although the stalk may also have some influence. Before grafting, stalk or scion, or both, are dipped in an auxin solution. This promotes an early union. The two main types of grafting are scion grafting and bud grafting. In scion grafting, the stalk may be only a root or a stump, and the scion may be a small shoot bearing one or more buds. Scion grafting is used to produce composite plants of economic benefits. The scion is obtained from a plant with superior characters, while the stock is derived from a plant resistant to diseases and pests and efficient in absorption of water and minerals. Scion grafting is further done in three ways: whip or tongue grafting, wedge grafting and crown grafting. In the first type, that is whip or tongue grafting, the stalk and scion, almost of the same diameters of 1 to 1.5 centimeters, are given oblique cuts of 5 to 8 centimeters long, followed by a notch in the scion. The notch is made in such a way that it exactly fits into the stalk. The scion is then inserted into the stalk and tied firmly. All buds are removed from the stalk but retained on the scion. In the second type of scion grafting, that is wedge grafting, the stalk and scion are of nearly the same diameter. A V-shaped notch is made in the stalk, while the scion is cut like a wedge. The scion is then inserted into the stalk and the two are tied firmly. In crown grafting, which is the third type of scion grafting, the diameter of the stalk is several times greater than that of the scion. Thus, many scions can be grafted on a single stalk. The lower ends of the scions are cut into long wedges and are inserted into the cuts made on the stalk. They are then tied firmly and the wound is sealed with grafting wax. The second type of grafting is bud grafting. In this type, the scion consists of only a single bud accompanied with a portion of the living tissue. A T-shaped incision is made in the bark of the stalk. The bark is then gently raised on edges and the scion is inserted into the incision. It is then firmly fastened with strings. The bud germinates after some time and becomes a part of the new plant. Roses, peaches and some other fruit trees are propagated by using bud grafting. Tissue culture techniques are now being used for effective vegetative propagation of several horticultural and agricultural crops. These techniques of propagation, which involve cell, tissue and organ culture, are collectively called micropropagation. In recent years, the technique of tissue culture is being increasingly employed to raise a large number of plantlets from a small tissue. The advantage of this technique over the conventional techniques is that in a relatively short time and space, a large number of plants can be produced. For micropropagation, axillary buds, shoot, apices, or any other plant part can be used as an explant. Virus-free plants can be produced if shoot, apical, meristems of about 0.25 millimeters long with 1 or 2 leaf primordia are inoculated. Explants are properly sterilized when collected from the field. Multiple shoots can be obtained by inducing callus. Plants produced in culture are carefully transferred to the soil. They are planted first in vermiculite and kept under high humidity for 10 to 15 days and then gradually taken to the greenhouse and can be transferred to the soil after 4 to 6 weeks.